Hello everyone. The subject of this video is covered in section 6 of chapter 4 of Essential Biochemistry, and here we review preparative techniques for separation of proteins. So how are proteins purified? And why would we want to do this? Well, we need to purify proteins because we want to determine the structure and obtain insights about the function of proteins. So that means that we have to isolate one protein that we're interested in from other proteins in the cell. We usually will rely on some unique characteristic that that protein has that other proteins don't have. In the past, if you were going to start out with the low-hanging fruit, you would choose proteins for their abundance. For example, if you had red blood cells, they're easy to collect, and about 95% of the dry weight of a red blood cell is hemoglobin. So that means that it can be fairly straightforward to isolate hemoglobin from a sample of red blood cells. What we do now, though, and this is for proteins that are not very abundant and would be very difficult to sort of fish out of the cell from all of the other proteins that are there, would be to clone the gene for the protein of interest and do some genetic engineering where you would take that gene and have some kind of a tag that will let you purify that protein later. And then you introduce that into the cell, and then you use a bacterial cell to essentially act as your a protein producing factory to crank out lots and lots of your protein. But it doesn't really matter if you're using the first example where you're trying to isolate a protein from a cell that actually made it, or from a bacterial cell. You're still in the position of having to isolate a protein that you want from other proteins that you don't want. And how we do that is the subject of this lecture. So what we would do if you would start out with the red blood cells is you would obtain and stabilize a solution of proteins from a cellular source. And then you'd fractionate the proteins to selectively eliminate the proteins that you don't want, and only the protein of interest remains. For bacterial cells, we have to do that as well. So what we call preparative techniques, what that language means is that the goal is to obtain large amounts of protein for future experiments, which means because we're doing future experiments, we want to preserve the biological activity. So we shouldn't do anything to our protein that's going to cause it to be denatured. Now keep in mind that large amounts might just be a couple milligrams. To a biochemist, that might be a large amount. But the idea is that you want to have enough protein at the end of your isolation protocol, and biologically active protein, that you can then go ahead and study that protein. So let's talk about some examples of preparative fractionation techniques that we can use. One is ammonium sulfate precipitation. So what we can do is we can add highly purified ammonium sulfate, which will salt out certain proteins as precipitates while leaving others behind in solution. And if we do that, we can separate some proteins from others by ultracentrifugation. So let's talk about how we would do this. Now suppose if you look over here on the left, I have a mixture of proteins, green, red, and yellow proteins, and I add some ammonium sulfate to that solution. If the red protein is the least soluble protein in that mixture, it will salt out, which means it precipitates. What I do at the same time, though, the yellow and the green proteins are more soluble, is I salt them in. So salting in actually increases the solubility of the proteins because the salt ions reduce the electrostatic attractions between the proteins. And that's shown here, where if I have this, if the protein is shown in these sort of brown circles right here, that if I add some salts, then the salts will interact with the charges on the surface of the proteins and keep the proteins apart, and that will cause them to be soluble. Now, if you get to a point where you add lots and lots of salt, then the salt ions will require lots of hydration spheres, and the proteins will eventually precipitate. So that's what happens here in this little diagram here, where I've salted out the red protein. If I add additional ammonium sulfate, I can salt out the green protein and leave the yellow protein in solution. So by doing a series of ultracentrifugation steps, I can pellet the red protein, then pellet the green protein, and then leave that yellow protein in solution. Now, suppose it's really the green protein that I want. That means I will throw away this tube right here, and I'll also throw away this supernamed. But what I'll have to do next is I'll have to take this green protein and then find a buffer that I can use to resuspend it. And then that takes me to my next step involving dialysis, because chances are your protein has ammonium sulfate associated with it. So if you add the protein to a dialysis bag, and dialysis bags have been manufactured to have holes that are small enough that allow the passage of ions, which are shown in this diagram as red spheres. So those can pass through the bag, and here you show there's an equilibrium where you can see that we have an equal concentration of these salt ions on either side. Whereas the proteins, which are shown as purple spheres in this drawing, stay inside. Now, it turns out if you do this, if you change your buffer, so for example, if I throw away this buffer and then place my bag into new buffer, I can get equilibrium to be established, and that will decrease the concentration of the ions inside the bag. And if you do that 3 times, pretty much you'll get rid of all of them. Okay, what's next? Chances are a lot of the proteins in our ammonium sulfate precipitations that we haven't just isolated one protein. There's probably a number of proteins there. So after we've removed the ammonium sulfate by a dialysis, we'll do chromatography to do further separation. So the chromatography experiments that we will do, well, like all chromatography, every chromatographic separation involves components of a mixture that are partitioned between the stationary phases and the mobile phases. And you as the experimenter need to be able to design your experiment so that the components of your mixture have different attractions to these 2 phases. So if everybody's attracted to the stationary phase, or if everybody's attracted to the mobile phase, you won't accomplish your separation. So in the biochemistry lab, we typically use columns. So I'm going to show some videos, and I'm going to not use the links, but I'm going to go to what I've already had queued up so that we don't have to watch ads. All right, where am I? I want to make sure I show the right one. Gel filtration. Okay, this is actually the video that's associated with my PowerPoint. So, and it happens to show ion exchange. So what's happening here is you have a sample mixture that you load on top of a column, and the column, there are beads that are microscopic, and I'm just going to pause this for a second. You load your sample on top. So the glass column with the beads, that's the stationary phase. That doesn't move. You have buffer, which flows through the column, and that's the mobile phase. So here's our sample mixture, and as the buffer flows through the column, we'll see what happens. The components of the mixture will separate, and we can collect fractions. So what's being shown here is that the red protein has a greater affinity for the mobile phase, whereas the blue protein has a greater affinity for the stationary phase, and the purple protein is in between. So I'm going to pause this again. So we can collect fractions, and in that way we can separate our proteins. Now, it's not likely that the proteins will actually have those colors. So what we'll need to do is we will need to monitor what comes off the column by using UV absorbance, because proteins will absorb light at 280 nanometers. So if I go back to this slide right here, tryptophan and tyrosine side chains will absorb at 280 nanometers. So if we take this effluent that comes off the column and pass it through an ultraviolet monitor, we can then interface that with a chart recorder, and it can show these peaks, and that's how we can identify where our proteins are located in which test tubes. So that's the general procedure. So what I want to do now is I want to show you the different types of column chromatography. So I have 4 types here: ion exchange, which is anion and cation, hydrophobic, gel filtration, and affinity. So let's talk about ion exchange first. So the beads that I mentioned that are in the column, these microscopic beads, in anion exchange chromatography, the beads are positively charged. Now, this is a source of confusion, or potentially could be. DEAE is diethylaminoethyl, and that's the functional group that's attached to these beads. It's positively charged. We call this anion exchange. It's because negatively charged things bind to it. So the chromatography isn't named for the bead, it's named for the thing that binds to the bead. So if I flow proteins down this column, the negatively charged proteins will bind to the beads and be attracted, but the positively charged proteins will be repelled, and they will elute first. So let me go back to my videos that I have queued up.
Ion exchange chromatography depends upon the reversible adsorption of charged solute molecules to immobilized groups of opposite charge. Most ion exchange experiments are performed in 4 main stages: equilibration, sample application and wash, elution, and regeneration. The first step is the equilibration of the stationary phase to the desired start conditions. When equilibrium is reached, all stationary phase charged groups are associated with exchangeable
So those are the beads right here.
such as chloride or sodium. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out
negatively
all
charged
material.
proteins are binding.
The sample buffer should have the same pH and ionic strength as the starting buffer in order to bind all appropriately charged proteins.
And everything that's flowing through here is positively
step,
charged.
Elution, biomolecules are released from the ion exchanger by a change in the buffer composition. A common way is to increase the ionic strength with sodium chloride.
So if you add sodium chloride ions, you can elute the proteins.
Relative to the number of charged groups on their surface. The final step, regeneration, removes all molecules still bound. This ensures that the full capacity of the stationary phase is available for the next run.
So let's go back to our PowerPoint. So what's being shown in the video is positively charged beads with negatively charged proteins. In this diagram, they're shown as negative red dots right here. The positively charged proteins will be repelled, and those proteins will elute first. In cation exchange chromatography, we use carboxymethyl groups. And these groups are negatively charged. So again, this is named for what binds to the beads, not the beads themselves. So positively charged proteins will bind and negatively charged proteins will be repelled. So at this point, you might be wondering how we know whether proteins are positively charged or negatively charged. Well, we have to use a concept called pI, which is the isoelectric point. That's the pH at which the protein is electrically neutral. Here's a whole series of proteins whose pIs are known. So proteins that are at the low end of the pI range have lots of aspartate and glutamates, or if I go down here to lysozyme, there's lots of lysine and arginines. So what you as the experimenter can do is, if you have some idea of the pI of your proteins, you can adjust the pH of your buffer solution. So if the pH is less than the pI, the protein will be positively charged. If the pH is greater than the pI, the protein is negatively charged. And if the pH is equal to the pI, the protein will be neutral. So you can control how what the charge of these proteins are, what the charges are, by controlling the pH of the buffer. So here's an example. So this is cation exchange, and here this shows negatively charged beads, and the beads are shown in gray. And this shows proteins flowing through the column, where the pink proteins have an overall negative charge, and they're repelled by the beads. So the pink proteins will come off first. The blue proteins are overall positively charged, so they will bind to the beads. So as it was said in the video, if you eventually want these proteins to elute, you're probably going to add some kind of positively charged ion in order to compete with the binding of those negative charges to get the proteins to elute. Usually what we do when we're designing our experiments is that the desired protein should bind to the bead. So in this example right here where cation exchange is being used, the desired protein is the blue protein. So the unwanted proteins will come off first and the desired protein will come off next. Okay, back to my list. How about hydrophobic interaction? Well, in this kind of chromatography, we have beads that are designed a little differently. They have either aliphatic or aromatic groups associated with them, so they are nonpolar. So in this way, you can separate proteins based on polarity. So let me go back to my videos that I have queued up here.
Separation in hydrophobic interaction chromatography depends upon the reversible adsorption of biomolecules according to their hydrophobicity. Most hydrophobic interaction chromatography experiments are performed in 4 main stages: equilibration, sample application and wash, elution, and regeneration. The first step is the equilibration of the stationary phase to the desired start conditions. Equilibration is performed by adding salt to the mobile phase. The general equilibration buffer in this step is 50 millimolar sodium phosphate with 1 to 1.5 molar ammonium sulfate. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material.
So these proteins are polar that are coming off.
Moderately high concentrations of salt.
And these
A
proteins are
concentration of
nonpolar.
salt that gives an optimal purification is chosen. In the third step, elution, biomolecules are released from the hydrophobic surface by a change in the buffer composition. A common way is to decrease the salt concentration of the buffer. Gradient elution can be used to separate the target protein from other bound proteins. Normally, a gradient of 10 to 15 column volumes is sufficient. The final step, regeneration, removes all molecules still bound. This ensures that the full capacity of the stationary phase is available for the next run.
Okay, so going back to our PowerPoint, again, this technique separates proteins based on polarity. The nonpolar proteins will have an affinity for these nonpolar groups, and the polar proteins will come off first. How about gel filtration? Sometimes this is called size exclusion. And I have a couple of videos here to show you. So here we have a mix, I'm going to pause this video, of big blue proteins and little red proteins. And the beads right here have little holes in them. So I think of them maybe as wiffle balls. So the large proteins can't get inside the beads, so they have no choice but to go around, and that's a faster pathway. Whereas the little proteins can go inside the beads, and that means they take longer to elute. So you can separate proteins based on size. So here's our matrix with the holes, and see how the red proteins can get inside the beads and the blue proteins are too big and they can't. So the blue proteins come off first because they go in between the beads, and that's a faster pathway. And we can collect fractions again, and then the pink proteins come off next. And again, we're going to interface the effluent coming off the column with a UV monitor because chances are our proteins aren't going to be pink and blue. So here are our fractions, and we can look at our chart recorder and see where our peaks are and figure out which test tubes we should collect for the protein that we want to isolate. Here's another video from the GE
Separation in
Healthcare.
gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed with a chromatographic medium, which is a gel. Molecules larger than the pores in the gel are unable to diffuse into the gel and are confined to the solution surrounding the beads. Molecules which range in size between the very big, excluded from the gel, and the very small can penetrate the pores to varying degrees based on their size. Access to the pores is limited by steric hindrance. If a molecule is smaller than the smallest of the pores in the gel, it will be able to enter the total pore volume.
So here we're separating based on size, with the larger proteins coming off first and the smaller ones coming off last. And that's shown in this picture right here, where we can see our gel matrix, and we can see that the blue proteins can't get into the pores, but the red proteins can. So the blue proteins come off first, and the red proteins come off next. Okay, so here's my list. Last type, affinity chromatography. So here you can, as the experimenter, you can make your own beads. It's actually not that difficult to do. If you have a protein that binds to a ligand, so ligand is a small molecule. This is not the best picture right here. I have a couple more that are a little better. But here your bead is attached to the ligand. And what that means is that if your protein binds to the ligand and the other proteins don't, you can get rid of the proteins that don't bind to the ligand. So in this picture right here, the ligand is this little black circle right here, and this red protein binds to the ligand, but the rest of them don't. So everybody comes off in one big peak initially. Here's another picture from another textbook where they show the bead and the ligand here are little sticks, and it shows that these red proteins can bind to the ligand because they have a binding site for it, but all of these other collection of orange proteins aren't able to bind because they don't have a binding site for the ligand. So what that means is that you have a big peak that comes off first, and then you have a smaller peak that comes off next. You'll have to change the buffer composition because usually what happens is the protein binds to the ligand so tightly that you'll need some reason to get it to come off. In this example, this is an enzyme called a nuclease, and it binds to thymine. So your bead has thymine attached to it. So the other proteins in the sample don't bind. So what you can do is you can elute it with a buffer that has high salt or higher pH or low pH, whatever is kinder to the protein, because remember, we want to preserve the biological activity. So let's go back to the videos that I have queued up, and let's look at another video on affinity chromatography.
Chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and dilution. The first step is the equilibration of the stationary phase.
So here the ligands are these rocket shapes.
The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all.
So the red and the green proteins don't bind, the yellow proteins don't bind, but the blue proteins do.
Non-binding molecules will pass through in the flow-through during washing with binding.
So here's your big peak that has all of those red, green, and yellow proteins.
In the third step, elution, biomolecules are released from the biospecific.
And here we're going to increase the pH to convince them to come off.
A common way is to increase pH of the buffer.
So some of you might have done this in your research. This is a very common type of affinity chromatography where if you are using the genetic engineering that I told you at the beginning, where if this is a plasmid, a circular piece of DNA, and that's codes for your protein, which is shown here in blue, and you can engineer a tag at the end. One of the ways to do this is to have 6 histidine residues in a row, because histidine will bind to nickel. So what you do is you can, once you've made your protein has these 6 histidines at the end, and you bind them to a nickel column, and then you wash the column, and anything that doesn't have those 6 histidines will come through, and then you usually add something like imidazole, which is just the histidine side chain by itself, and that will elute the protein. So, as to not give you the idea that you just have to do one step and that's all you have to do, um, typically it takes a couple of steps to purify a protein, especially from a natural source. So suppose I do ion exchange chromatography, and this is what my chromatogram looks like. There's not really any clear peak. So I collect all these fractions and say it's an enzyme. So what I do is I have to go through every single one of these tubes, and I have to do an enzyme assay until I find my protein. So I have an assay, so there it is. So I don't know, maybe it's this peak right here. So I pull these fractions, and then I will do another step. I'll do gel filtration. And again, I don't have a clear peak right here. So I have my large proteins to small proteins. I take all my fractions. I do an enzyme assay on every single one. And there's my protein right here. So I grab those test tubes. And then now I load them on an affinity column, and I have a ligand that's specific for this protein. And all of the proteins that are left that don't bind to the ligand come first. And you can see when I do my enzyme assay, I have a pure protein. And the other way you can tell is you can run a gel. So if this is lane 1, if this shows all the proteins that I started with, and this is lane 2 that shows it's still a little bit impure, and here I am at the end and I have a single band, that tells me that my protein is pure. But that involves affinity techniques, and that is the subject for the next lecture. So until next time, do the Chapter 4 assigned problems in Essential Biochemistry. You can look at the Instant Notes, section K1. These are my references. Thank you for your attention, and I will see you in class.